Thank you all for coming, joining us today for this talk. This talk is part of the Nathan's Lab 30-year reunion, and we're happy to have many of our past members here with us. They all came from far and near. Regrettably, not everybody could make it. And this reunion is to celebrate 30 years of joint work in science with its exhilarating highs, when your favorite hypothesis survived a grueling experiment to unravel a new understanding of biological process. And then, even more often, there are the lows when the critical experiment kills your pet hypothesis and sends you back to square one. But we are always striving for fact-based truth. And then there's, of course, funding and funding and funding. And to share his perspective on, I think, these matters and much more, let me introduce the one who needs no introduction here, our group leader and my colleague and friend for many, many years, Jeremy Nathans.
Well, thank you all for coming. It's a great pleasure. Thanks, Amir, for that lovely introduction. As Amir said, it's our 30th birthday, and we're delighted to celebrate it with you. So first, I want to have a special thanks to Chris Cho. Chris organized everything. Here's Chris. He's consulting with Joe Biden here about the Cancer Moonshot Project. And Chris, in addition to being a remarkable scientist, is also a ballet dancer. He's heading back to medical school. And where's Chris? There he is. And Chris now realizes that if medicine doesn't pan out, he has a second career waiting for him as an event organizer. Okay, now if there's any theme here, it's this one. Biomedical research is the ultimate team sport. And I'd like to acknowledge the many depths of gratitude to the many, many team members. First, of course, the Nathan's Lab, which has evolved over many years. Here we are about 3 years ago at Yan Shuang's house. But of course, the team is much, much bigger. We've had remarkable leadership at every level in the school. In the Department of Molecular Biology and Genetics, Tom Kelly and Carol Greider have been the chairs while I've been here for 30 years. In neuroscience, we've had Saul Snyder and Rick Huguenier. Rick is here. And in ophthalmology, we've had Mort Goldberg and Peter McDonald. Just extraordinary leadership, extraordinary wisdom, and that, of course, makes all the difference. But of course, the team is much, much bigger. So those who know me know that I carry around a little book, a little black book. And in my little black book, I have a list. The book's in here somewhere, well, wherever. Now I've got my equipment. Anyway, I have thousands of names, actually about 2,000 at last count. And about 1,000 of those are Johns Hopkins people in the book. And so here's just a typical page. This is from the Animal Services Department. So I just write down everyone I meet, the people who change the cages. Alan Gordy is our current cage changer. And nothing would happen without all of the support people here at the university. Of course, there have also been many support people in the lab, so I just want to mention the administrative assistants. There was a time when we absolutely required that you have a first name of Terry. The occasional exceptions were made. Human resources would eventually going to crack down on us on this. But of course, there are many other people who've played essential roles, really the backbone of the lab, the students and postdocs and technicians and research associates. Here's a list on a timeline of all the graduate students who've worked with me, all the postdocs who've worked with me, and then here are the scientists and technicians. I include myself in this list here, and I just want to note there are some long-timers here. Yan Shuang was one of my first graduate students, and she and I have worked together for 30 years. Half my life and more than half of hers. Phil Smallwood, Amir Ratner, who you just heard from, and John Williams have also been long time with me. And so the five of us at the moment, we're kind of the survivors on this island here. The five of us are still together. It's going to be sort of a geriatric lab pretty soon, but that's all right. Now, I want to say two members of the family are no longer with us. Nupur Thekti and Genshi Shea died much too young. They were wonderful, wonderful lab members, and we miss them very, very much. And of course, a part of the team, an essential part of the team, is those who invest in us, those who put their confidence in us through their financial support. These range from the large and impersonal, the National Eye Institute, to the very personal Mr. Labovitz, who lives in Washington, D.C., has just decided on his own to support our lab. And we're very grateful to all of them. And I'll mention in particular, Peter McDonnell arranged for an anonymous donor to endow a professorship for me, the Samuel Theobald Professorship. And we're very grateful to Peter and to the donor. Now, I want to mention, this is beyond the team, but I want to mention the importance of mentors. Obviously, everyone benefits enormously from mentors, and I've been extremely lucky over the years. My trajectory in science has been influenced enormously by the mentors that I've had, and I just want to mention three of them. David Hognes, who was my thesis advisor at Stanford, here he is in his office about 35 years ago. For the youngsters in the audience, there's this funny object in the background with this round thing in the middle. You can Google it, figure out what it is. Dave was wonderful, a remarkable man and remarkable scientist. Lubert Stryer, who's shown here, you may know the name. He wrote the best-selling biochemistry textbook, another mentor and just wonderful friend. And of course, my father, Dan Nathans, with whom I overlapped here for about a dozen years. And of course, not only was he a great scientist and mentor, but it was great to be in the same town with my parents. And here's just an example of the pleasures of living close to one's parents. This is my daughter, Riva. She had a long day. She's taking a snooze here. Now, in terms of science, let's focus on science for a second. I think it's fair to say the lab trajectory really began at Stanford when I was a student. So I was working in Dave Hognes's lab, essentially trying to find a thesis project, and it was a bit of a challenge. I was initially working on a project that was clearly going nowhere, and so was open to the idea of something new. And I heard a lecture from Dennis Baylor, an electrophysiologist at Stanford. Dennis was King Yao's postdoc advisor, and Dennis and King, this is in the late 70s, early 80s, had developed this new method of recording from photoreceptor cells. So this is a little chunk of frog retina, and these objects sticking out are the outer segments of the rod photoreceptors, and one of them has been drawn up into this glass electrode, and here in yellow is the exciting light beam. The rest of the image is bathed in infrared light, and this beam of light is exciting that rod outer segment. And this electrode configuration invented by King and Dennis revolutionized the field of retinal physiology, and it allowed them to detect, among other things, the responses to single photons. And I heard about this when Dennis Baylor gave a lecture around 1980, and just about the same time, within a month or so, Lubert Stryer also gave a lecture on vision, and in it he described his discovery of the G protein cascade for vision. That is, rhodopsin is a G protein coupled receptor, not appreciated until that moment, and it activates a cascade of biochemical events that's very much like the events that occurred during hormone signaling, for example, when the beta-adrenergic receptor is activated. Well, those two lectures electrified me, and I realized that I had to work on vision. I had to work on the retina, and I've never looked back. The retina is incredibly beautiful. It's beautiful physically, as shown in this diagram. This was drawn in 1887. Amazingly accurate. Rods and cones at the top, the central layer of neurons in the middle, and ganglion cells at the bottom. It's beautiful at every level. This is an electron micrograph, just colored to make it look pretty, of the outer segment, inner segment junction of a photoreceptor. These beautiful layers of membranes are the outer segment that are chock full of the visual pigment, the light sensor, And here are the mitochondria that power the whole apparatus. And I was fascinated by this. And so I began working on vision, and as a student, I isolated the genes that code for the light-sensing proteins, the rod and cone pigments that mediate dim light and color vision in humans. And then when I got to Hopkins, basically one thing led to another. So we started out working on visual pigments and color vision, very quickly branched out into inherited retinal disease, thanks in no small part to my colleagues at the Wilmer Institute, who really were enormously stimulating and influential in this work. We decided to start working on retinal development, and in particular on a family of transcription factors called POW domain factors that control ganglion cell development. And then in the mid-90s, we branched out yet further and started working on a family of receptors called frizzles, which turn out to be master regulators of embryonic patterning, CNS development, and vascular biology. And they continue to fascinate us to this day. Now, hand in hand with the biology has been technology. And it's a dance. It's like Ginger Rogers and Fred Astaire. You can't have one without the other. And we have been moving progressively, expanding progressively out from our very narrow start point in molecular biology and human genetics to include spectroscopy, mouse genetics, various genetic technologies for neuronal labeling, more recently computational biology in many of its forms. Of course, this wasn't done in isolation, especially the technology part. A lot of it was driven by what was going on around us. And I should say, as a molecular biologist, there has been no better time to be alive than now. Because if you simply ask, and this is a very incomplete list, what has been going on in the last 40 plus years, it's amazing. Gene isolation, DNA sequencing, PCR, the whole computational part, automated Sanger sequencing, knockout mice, on and on and on, with no end in sight. And there are two aspects of this that I especially want to mention. Two revolutions, I think, in our scientific lifetimes that have really changed everything. The first, of course, is computing. So this is what high-end computing looked like when I arrived at Hopkins in fact, January 1988. Here's the Apple that had 2 or 4 megabytes of RAM. That was hot. And this is what DNA sequencing looked like. Actually, it didn't look this good. This is probably my best gel ever. But you had to start each day by washing your gel plates. You put the gel plates together. You pour the acrylamide. You run your reaction. So around about lunchtime, you might think about doing a real experiment. But this has all changed enormously. And here's what it looks like now. This is an order form where you just send over your sample and you type in a few numbers, including numbers that have dollar signs in front of them, as it turns out. And then a week later, 400 million reads come back. So to the students in the audience, let me tell you, you got it good, because it wasn't so easy in the old days. Now, let me describe a series of snapshots, little vignettes of some of the work we've done. It's very selective. I don't have time to give you the real story, the complete story. That would be a multi-hour lecture. It would be the Fidel Castro style presentation. Let's not do that. But I'll just give you a flavor of some of the things we've done over the years. So here's a general area of interest: visual pigment structure, function, and genetics. So visual pigment means the light sensors in rods and cones. And one very beautiful set of experiments was done by Shanath Merbs, who's here in the audience. And Shanath was an MD PhD student in the lab, and she succeeded in producing, as recombinant proteins, the apoproteins of the human long, medium, and shortwave sensitive pigments, reconstituted them with their chromophore 11-cis retinal, and took their absorbance spectra in a spectrophotometer. And here they are. The long wave sensor, the medium wave sensor, and the short wave sensor. And these are, to this day, the most accurate determinations of these spectral sensitivity curves which underlie all of human color vision. And when Shanath was doing this work, she made a very interesting observation, which was that 2 allelic forms of the long wave sensitive pigment, one of which has an alanine at position 180 and occurs in about 40% of humans, or I should say 40% of X chromosomes, because it's X-linked. So they're heterozygous females who have one of each. The other has serine at 180, and that's the other 60% of X chromosomes. but males have either alanine or serine, and those differ by about 5 nanometers in their absorbance spectrum. So in this room, there are plenty of people here who believe they have, quote, normal trichromatic color vision, but in fact they disagree subtly with each other in terms of their color matching because they have one or the other of these allelic forms. Perhaps the most common allelic variation in our species that affects a psychological property. And then Qinghua Sung began working on rhodopsin mutations that cause retinitis pigmentosa, one of the really significant inherited retinal dystrophies. And she and I discovered a number of mutations, and there was a whole class that especially got our attention because they were clustered at the extreme carboxy terminus of the protein. So this is a G protein coupled receptor. The extreme C terminus is in the cytoplasm. And it wasn't at all clear what these mutations were doing. These were single amino acid substitutions, for example, within 1 or 2 or 3 or 4 amino acids of the carboxy terminus. When we made the mutant proteins in transfected cells, we saw that the yield was normal. They bound the chromophore normally. They absorbed light normally. They activated the G protein. They interacted with a kinase that phosphorylates the protein and so on. They went to the plasma membrane. But it occurred to us there was one thing that they were not modeling in this transfected cell system, and that is the movement of the protein to the photoreceptor outer segment, the specialized appendage that photoreceptors have that has all the phototransduction machinery. And we thought maybe the C terminus was actually the zip code for directing the protein to that outer segment. And that turned out to be a correct guess. And here's the proof of that. Qinghua made a transgenic mouse in which one of these mutants, and this one happens to be a truncation mutant, and she picked that one, very clever choice, because it's missing the last 5 amino acids of rhodopsin, so it's truncated at the 344th amino acid. And therefore, she could use a synthetic peptide corresponding to the mutant C terminus and raise an antibody to that peptide, which sees only the mutant protein and not the wild type protein. And so with the mutant-specific antibody, she could detect the mutant protein alone in a section of retina, and with an antibody against which we had, against the extreme C terminus of wild type rhodopsin, we could detect the wild type protein alone. And this is immunostaining in the same tissue section. And what we see is that in this section of retina, so here's the Nemarsky image, the wild type protein is correctly trafficked to the outer segments, which are in this layer here, and the mutant protein is inefficiently trafficked. And because it's stained in the same section, we know it's not that the cell is unhealthy and therefore the trafficking is screwed up. It's a defect in the protein itself. Now, at about that same time, Yanshu Wang and Don Zak were looking at the expression of cone pigments, and they realized that a little region upstream of the long and medium wave pigment genes, and we call this the locus control region, it's basically an enhancer, is required, absolutely required for expression of those genes in cone photoreceptors, and that's shown here with the beta-galactosidase transgene. This is in mice. So here's cone photoreceptors, individual cone photoreceptors stained with XGAL. And as a result of that work, we eventually realized that that little locus control region pairs with either the long wave promoter or the medium wave promoter. And it's that pairing which determines the choice of a cone to be either a long wave cone or a medium wave cone. It's basically a toggle switch. It can either be one or the other, but it can't be both, because that LCR can only accommodate one promoter. Now, another little vignette. Here's one on retinal development, physiology, and disease. And here I just want to concentrate on one particular aspect of this large story, which is the cycling of retinoids within the retina. So there's a constant movement. It's happening right now in your eyes. And it's happening all the time, pretty much. Constant movement of the chromophore of the visual pigments. Here it's shown, it's 11-cis retinal. It's joined covalently to an amino group, an epsilon amino group of a lysine. So this is what rhodopsin looks like and what all the cone pigments look like. Obviously, the protein part is not shown here. But this 11-cis retinal is the dark state. And when it's struck by a photon, it isomerizes to all trans, and it dissociates eventually from the protein. So the dissociation allows the protein to be recycled back to the dark state. It needs to get a 11-cis retinal reattached to it. And how does that happen? It's rather involved. the retinal that's released is very quickly reduced to retinol. That's the alcohol. So this is an aldehyde. There's an alcohol. There's virtually no free aldehydes in biology, basically. Aldehydes are reactive with free amines. It's bad news. They're generally toxic. So the way to make this compound safe is to convert it to the alcohol. And this enzyme, which is called retinol dehydrogenase, actually named oddly for the back reaction. So it dehydrogenates retinol to retinal. Enzymes, of course, can go in either direction. Was known since the 1930s, but it was known as an activity, but it had never been identified. No one knew what that protein was that was the retinol dehydrogenase. And then the retinol is shipped to the pigment epithelium. It's esterified onto a fatty acid. It's reisomerized to the 11 cis configuration, converted back to the aldehyde, and shipped back to the photoreceptor cell. Okay, so around the year 2000, Amir Ratner, who gave the introduction, Amir and I decided to look for this enzyme, and Amir, through a very, very clever series of sort of bioinformatic approaches, found it in a collection of sequences that we had produced of cDNAs from human retina. And here's just an immunostain that Amir did. This is a retina section, and you can see in brown is the immunostaining. It's only in the outer segments. Here's a no primary antibody control. And when we produce the recombinant protein, here's untransfected 293 cells. We're monitoring the reaction spectrophotometrically, and I should just say the retinal and retinol have different absorbance spectra, so as the reaction proceeds, the spectrum changes as substrate turns into product. So it's easier to follow the reaction, and here it is in the transfected cells, and it absolutely requires NADPH, and it doesn't use NADH. That was a signature of this particular enzyme, and you can follow it over time and do the usual enzyme kinetics. So we found the critical enzyme. Now that was, in a sense, something that was known. It was sort of a known unknown. But the next part of the story was an unknown unknown, as Don Rumsfeld famously said in an infamous context. And that was, again, something that came out of the collection of sequences. So I'll just say parenthetically, when Applied Biosystems put the first automated sequencer on the market, and Hopkins got one thing we did very quickly was realize this would be great for sequencing little snippets of cDNA and figuring out which genes are expressed in a given tissue, sort of the precursor of RNA-seq. And so we did that with human retina. We collected about 5,000 sequences, basically ran out of money at that point. And one of the things that came up in that collection was, and I should say we dumped it into GenBank, so it was sort of a public resource for everybody. One thing that came up and that we had not sort of picked up on, but we were alerted to by a group at NCI, was that there was an ABC transporter that was highly enriched in this collection and was not found in any other collections. And this we initially called ABCR, ABC Retina, as a transporter. And this was Mike Deans and Rando Alekmetz, who were ABC transporter experts, who contacted us about this. And not only that, they had mapped it to a chromosome, and they realized that it mapped very close to the place where a human disorder called Stargardt disease maps. Now, Stargardt disease is the most common early onset form of macular degeneration. Occurs in about 1 in 4,000 humans. It had not been identified, but its map location was roughly known, and that similarity in location led Alekmetz and Dean and our lab and two geneticists, Jim Lupsky and Mark Leppert, who had been on the trail of Stargardt disease, to all work together, and very quickly we found that the ABCR gene was in fact the Stargardt disease gene, so those people have mutations in this transporter. And then Hui Sun asked, where is the transporter in the retina? And what he saw was by immunostaining, so here's a known retina outer segment marker, the cyclic G-gated channel, and here in red is the transporter. It's only in photoreceptor outer segments. And when Hui looked at higher magnification, this is the early days of confocal, what he saw was the channel was in the plasma membrane. Let's just look at this particular outer segment here at the surface, and the transporter was in the interior. You can see when the two are melded together, the green is outside of the red. That is, this is a transporter that's inside of the outer segment. It's not at the plasma membrane. It's not transporting something across the cell's surface membrane. It's transporting something inside the cell. What could that be? And we guessed it might be retinal. It might be involved in the retinoid cycle. That turns out to be right. I can't give you all the details, but the bottom line is when retinal is released from the photoexcited pigment, it needs to be basically picked up by the transporter and presented to the cytoplasmic retinol dehydrogenase for the reduction reaction. And here's just a very beautiful experiment that we did where we looked at the kinetic properties of the transporter. So we tested many dozens of compounds as potential stimulants of the ATPase activity of the transporter. Why do we do it that way? Because ATPase, setting up an enzyme reaction with ATPase is very straightforward. It's easy to measure ATP hydrolysis. It's very hard to measure physical movement of a small molecule from one location to another. That's a very difficult reaction. And besides, we wanted to have a series of test compounds. We would have to set up a separate transport assay for each one if we were going to do it that way. So instead, we flipped the system on its head. We said, well, let's just monitor ATP hydrolysis and ask which compounds stimulate the ATP hydrolysis. So like many motors, this motor also runs all the time. So it's a little bit like when your car is in neutral. You're still using up gas, even though the car is not moving. And the same is true here. So just look at the open circles for a second. If we plot the data on a double reciprocal plot, so it's 1 over ATP concentration on the horizontal axis, and it's 1 over initial velocity of the reaction on the vertical axis. For those grad students in the lecture hall, you probably had to remember this for your oral exams. And forgive me if this is bringing bad memories back, but remember, this is Michaelis-Menten kinetics here, and it's all true, and every enzyme obeys it. And so, of course, if you just follow this for a second, if we go towards the 0 point, that's 1 over ATP getting closer and closer to 0. That means ATP concentration is increasing, right? ATP going up to infinity is the 0 point. And if we just look at this basal reaction, we can ask, how does that set of data points change if we add retinal to the reaction? And the answer is it shifts. It shifts in a parallel manner. I don't have time to go into all the details, but this parallel shift is the enzymatic sine qua non of a coupled reaction. If you have product A going to B, coupled to product C going to D, then the effect of C going to D on A going to B is exactly this parallel shift. And so what we inferred was these are coupled. There's some sort of coupled event. As ATP is hydrolyzed, retinal is being moved. That was the inference. And that led us to a model. This is now the accepted model for how ABCR, now called ABCA4, works. It's in the disc membrane, a little bit of which is shown here. Here's the plasma membrane. And as rhodopsin, shown by these little balls, releases its retinal, the retinal is transported by ABCR to the retinol dehydrogenase and then eventually shipped out. And in Stargardt disease, where the transporter is defective, the retinal accumulates inside the photoreceptor outer segments. It's eventually engulfed by the RPE cells, and this leads to the accumulation of toxic retinoid derivatives. And that's the pathophysiology of the disease. Okay, a second little vignette, or a third. So in the 90s, the mid-90s, again, thanks to this high-throughput sequencing approach, this random sequencing of retina cDNAs, we discovered the frizzled family of receptors. And these are receptors that look like G-protein coupled receptors. They have 7 transmembrane segments, but it turns out they do many different things. And as far as we know, they're probably not G-protein coupled, although that's still debated. But our first paper on this family, Yan-Shu Wang, here's the first author, published in JBC, we speculated at the end about what they might be doing. I'll just read this to you. I'll read it partly because every now and again your speculation is right. So this is the right one. I can't resist. So what we said was, if frizzled proteins act as receptors, that would imply the existence of a corresponding family of ligands. In the simplest case, if there's one ligand for each receptor, then mammals would be expected to have at least 8 frizzled ligands, because at this point we isolated 8 receptors. Actually, there are 10 of them in the genome, as it turns out. And zebrafish at least 11. Currently, the only family of ligands known to be of this size and for which no receptors have been identified are the Wnt proteins. And it turns out there are 19 Wnt. The availability of a large number of frizzled genes should facilitate a biochemical test of the possibility that these two families of proteins interact directly. And within 6 months, we had, in collaboration with Roel Nusse, shown that this was correct. So Zhenqi Xie and Purnima Banot and folks in Roel's lab, and Roel was sort of the expert, is the expert on Wnt, had put 2 and 2 together and shown that the Wnt actually bind specifically to the frizzleds. And so at that point, frizzles weren't looking for any new ligands, but then we got a bit of a surprise. So in the early 2000s, or really starting in the late 1990s, we decided that the way to tackle the frizzled family was simply to make knockout mice and let the mice give us the answer. So we're not going to try to guess too much anymore. That first guess was good. We're batting 1,000. Let's stop there. And let the mice do the talking. And so we made regular and conditional knockouts, and one of them, frizzled 4, was especially interesting. And what Yan Shu discovered is that if we look, and it was especially gratifying because it took us back to the retina, and many of the frizzleds are involved in biology all over the body, so it wasn't necessarily going to be a retina-centric direction here. But it turns out that the wild-type retina, shown here with the blood vessels highlighted in red, has a very stereotyped vascular tree. There's an inner tier of vasculature here on the vitreal face of the retina. There's a capillary layer on one side of the outer nuclear layer and a capillary layer on the other side. It's always that way, and it's exactly that way in us. In a frizzled 4 knockout, the vessels form on the inner face of the retina, but then as they try to invade, they can't make a capillary plexus. They just form these little balls of cells. And that really caught our attention because it was an exact match for a phenotype that was already in the literature, and that was the Norrin knockout. NDP is the name of the gene. But this is an X-linked gene in us and in mice, and in humans, mutations in this gene cause Norrie disease, a severe blinding disorder in which it's about 1 in 50,000, in which the infant is born blind. Because it's X-linked, it's a male infant. But I think you can appreciate the essential identity between these two phenotypes. Now, what made us even more excited was that the Norrie disease gene codes for a protein, Norrin, which looks like a ligand. It's got a signal peptide. It's a far-flung member of the TGF-beta superfamily. And since frizzleds obviously are receptors, the logical possibility was that Norrin binds frizzled 4. And Chung Zhu tested that idea and found that it was correct. Not only does it bind, it also activates canonical Wnt signaling. So even though it's not a Wnt, it has no homology at all to Wnt's, it acts exactly like a Wnt. And so here's just a binding assay. Here's the frizzled 4 ligand binding domain expressed as an IgG fusion, nice binding curve. Here's a different frizzled, shows no binding. So it has nanomolar binding. I should just say, Taoxin Chang, who's here in the audience, as a graduate student, solved the crystal structure, the co-crystal structure of Norrin bound to the frizzled 4 ligand binding domain when he was a student at Oxford University. So the story has really come to a very satisfying point. But it's gotten more interesting and more complex. It turns out that canonical Wnt signaling doesn't just control angiogenesis in the retina, it also controls angiogenesis in the brain and in the spinal cord. And furthermore, the entire signaling system is used afterwards and throughout life to control the blood-brain barrier and the blood-retina barrier. So canonical Wnt signaling does 2 very different things. And in the brain and spinal cord, it uses not only Norrin signaling, that is the ligand Norrin and frizzled receptors, but it uses Wnt's as ligands, 2 Wnt's in particular, 7a and 7b, also uses frizzled as a receptor. And it uses a couple of absolutely critical co-activator proteins that have been the subject of interest in our lab for a number of years. And I'll just show you one piece of data in this direction. Here's an example of what a mouse embryo looks like if it's wild type. This is at embryonic day maybe 13, I can't remember. Here we see the vasculature in the cerebral cortex. We'll just focus on this particular place, although there are other places that show the same story. And here is one of these co-activator proteins. It's called GPR124 that has been knocked out in this embryo. There's a lot of bleeding in the brain, a little bit in the spinal cord. And if we look for the vasculature, it's missing. So it's the same angiogenesis defect that we had seen before in the retina, basically. And if we simply flood the brain with norrin, the ligand that the retina uses, it completely fixes the problem. So this is a brain that just needed, or the vasculature just needed canonical Wnt signaling. It doesn't particularly care what ligand it is, as long as the blood vessels get that signaling. And, oh, actually, this was wild type, pardon me. That's wild type. This is the mutant. That's the wild type control. That's the mutant rescue. Okay, so that is still, I'd say, an active area of research in the lab. We're still not at all clear how it works biochemically. How do these different proteins work together? How do they mediate the specificity of signaling? And we'd like to get those answers. Okay, another vignette. Frizzles do many things. In addition to canonical Wnt signaling, which is basically a gene expression program, they control the polarity of cells in tissues, especially epithelial sheets, where polarity is critical for the function of the sheet. So they're basically like the components of a compass, where every cell has a little vector within the plane of the epithelium. And here's just an example of how we've inferred that. If you look at the skin of a mouse, a wild-type mouse, the hairs are all essentially parallel. They go from nose to tail. And here is the skin of a mouse schematized with arrows showing the direction of hairs. If it was a wild-type sample, not shown here, but if it were a wild-type sample, all the arrows would be going from the nose to the tail. This is where the ears were, and these are where the eyes were. Okay, so we have a lot of these. It's like a little Daniel Boone's cabin here, lots of little pelts. But this is a mutant where frizzled 6, one of the frizzleds, is inactivated. And this is work from Nini Guo and Yanshu Wang, and computational work from Tudor Badia. And what Nini discovered, and Yanshu and I have continued to work with, is that you get these very unusual patterns. The orientations can be reversed, but they're not, in the adult case, they're not completely random in the sense that they self-organize. So although the orientations of the hair follicles are inappropriate, they're communicating with each other, even though the frizzled 6 gene is mutant, and they're organizing themselves into these macro patterns. You can see some of them are whorls, some of them are crosses, and so on. And epithelia throughout the body are using this system to organize the orientations of their constituents. So just to give you a sense of how broad it is, in the fallopian tubes, and we all wouldn't be here if it weren't for this system working, as you know, the egg emerges from the ovary at the tip of the fallopian tube and has to make a very long journey down to the uterus. How does it do that? Well, it turns out the uterus has motile cilia. It has epithelial cells where the cilia are always pushing in the same direction. And they push the egg from the top all the way down to the uterus. That system uses this planar polarity system. If you mutate the planar polarity genes, those cilia are misoriented and they just beat randomly. And the same is true for the cilia in our pulmonary tree that move the mucus from within the alveoli up and out. And the same is true for the cilia in the ventricles that move the cerebrospinal fluid around. All those things are oriented, and they're oriented by the polarity system. And if you look, and this is Yanshu Wang and Nini Guo's work, if you look at the locations of the proteins shown in green with respect to cells in epithelia, it's very striking. They're asymmetrically located. So this is the inner ear. The red indicates the outline of one of the hair cells. Here's another one. And the proteins are localized to just one side of the cell. Okay, now along with all the biology, we've been fascinated by technical development and have tried to make our own little contributions. And the one place where I think we have made some contributions is in the realm of mouse biology, mouse genetics, mouse analysis, and in particular in imaging. So anyone who works on the retina, of course, is impressed with the beauty of the structure and with the facile way in which it can be imaged. Why? Because it's basically a planar object. It's a thin object, thin enough to be imaged in its entirety so that you don't have the problem of sectioning artifacts and wondering whether you're missing pieces here or there. And so here, for example, is a flat mount where we're looking at the vasculature just color-coded by depth. So blue is the most superficial, and then green is in the middle, and red is deepest. And not only can you see it in little regions of retina, you can see the entire retina. So here's a flat mount. This is one of Amir's, just showing a mutant retina in which the vasculature is stunted in its growth, and we can see a compensatory vascularization from the hyaloid vasculature. And that idea of two-dimensional imaging really captured our imagination. And as it turned out, since the frizzle business led us to think about polarity in epithelial sheets, and in particular in the skin, we got to thinking, why can't we treat the skin the same way we treat retinas? The skin, after all, is also a two-dimensional object. And so Hao Chang and Yan Shu and to a lesser extent me have exploited this. And so here's an example. This is Hao's handiwork. This is actually the tail of a mouse. The head would be here. The tail would be out. The end of the tail would be here. The skin of the tail has been peeled off and unrolled and flattened. And what we're seeing are little clusters of 3 hair follicles. They come in triplets. There are 3, there's 3. And you can see it's essentially a crystalline array. Very beautiful developmental system. Here's something that Hao Wu did. Again, with the skin as the object of interest, we asked, what if we flatten it and we visualize with a very sparse labeling technology, and this was developed by Tudor Badia and Tom Rotolo, so we can label, say, 1 cell out of 1,000. What if we visualize the nerve endings that are in the skin? What do they look like? This had never been done before. The skin analysis had always been done in cross-section, and the nerve endings, of course, are huge. So very large arbors. It's like taking a cross-section through an oak tree. You don't get a sense of what the full tree looks like. Here, because the skin is intact, we can see the arbor of a single nerve cell, and each of these circles is a hair follicle. So this one wraps around hair follicles. The total length of the axons from this one cell is longer than the size of the mouse. So it's a very elaborate axonal arbor. And here's an example from Lucas Hua. This is a section of a whole mount of a mouse embryo stained for neurofilament. Again, color-coded by depth. You can see all the nerve fibers. I should say this one has really captured the imagination. At latest count, it's on the cover of 2 textbooks. And it's really an iconic picture. And then again, Hao Wu has exploited mouse genetics to look at exon activation. So what we've done, and this is with Hui-Min Yu and Phil Smallwood as well. And Hui-Min and Hao and Phil made this pair of mice in which one X chromosome had a green reporter, the other X chromosome had a red reporter. And when we cross them and ask in females who have one X of each, where are the red and green cells, we could basically track exon activation, which, as you know, is a stochastic process and is variable. And what we observed is a remarkable degree of position to position and animal to animal variability. And we especially were interested in the brain. And here's just an example, a coronal section through the brain. And you can see that this side of the cortex has many more cells that have activated either the maternal or paternal version, I don't know which is which, compared to this side, which has activated more of the other version. So this is a level of genetic diversity, somatic genetic diversity, which is unique to female mammals, unavailable to male mammals, and I think that may explain a lot, as we're seeing in today's politics. Okay, enough. I better not say any more. It'll be my Larry Summers moment. But it's fair to say that women's brains are more complex at the level of X chromosome expression than our male brains. I think that's a true statement. Okay, genomics and epigenomics, and now we're getting more towards today, and Alyssa Moe really, with Amir, spearheaded bringing this into the lab, and here's just the latest from Mark Sabasso. We've been fascinated by what next-gen sequencing technology can do. Here's an example, a collaboration with Joe Ecker's lab at the Salk. And so what Mark has done is FACS sorted with an appropriate transgenic mouse vascular endothelial cells that are in the liver or the lung or the kidney or the brain. Now it turns out the vasculature is not just a bunch of piping. The piping is customized to each organ in which it resides. And what Mark has shown is that the gene expression patterns for each of these different organs is distinct. The gene expression patterns of the vasculature is distinct. And you can see that here's just one example. This is a gene, MFSD2A. It's expressed in brain vasculature exclusively. And in dark blue is brain, light blue is liver, pink is lung, and green is kidney. And what we see here is the methylome pattern at the top. So this is the locations, the density of CpG methylation. This was done with Joe Ecker. And we see differences. For example, this region is hypomethylated in brain, region over here. And down at the bottom, these 4 tracks represent the accessibility of the chromatin as determined by this method called ATAC-seq. Again, this is all whole genome. And what you see is brain-specific accessibility, brain-specific accessibility, brain-specific accessibility, exactly in regions that are hypomethylated. And this has led Mark to a systematic analysis of the sites for transcription factors, the identities of transcription factors, and essentially the regulatory network that imparts the organ-specific properties onto the vasculature. And that's an ongoing story. And finally, the last science story, which is just really beginning, pioneered initially by Xiaowu Gu, who was a postdoc in the lab, and now, and with Phil Smallwood, and now in the hands of Tao-Shin Chang. We're interested in the structural biology of proteins that are important for vascular function. And so we've got our first crystals, and I should say Jennifer Cavern in School of Public Health and Sandra Gabelli here in the School of Medicine have been absolutely essential in getting this going. And we're excited to pursue this with a number of targets of interest. Okay, let me just say some things about projects that haven't worked. There are many more of them than the ones that worked. So this talk, in a sense, has been a fiction up until now. I'll just describe 2 very briefly. One that has fascinated me for decades is human sexual orientation. So this is an absolutely fascinating variation in human behavior that very likely has some biological component, and the biological component is unknown. Anything that has a biological component, I would argue, very likely has a genetic component. The genetic component is also unknown. And we've been working on this off and on for 30 years, and we've managed to publish only 1 paper, and that was 25 years ago. Pioneered by Jenny Mackey, who was a technician in the lab at the time, and it was a negative finding. Sequence variation in the androgen receptor is not a common determinant of male sexual orientation, despite the fact it has been very well cited. But it's been a lot of fun, and here's Phil and me at the Gay Pride Festival in Washington, D.C., advertising our wares. We were collecting cheek swabs to do genetic analysis. Got a lot of swabs, got a lot of interest. I think it didn't help that the booth next to us was giving out free cookies, so there were people around. And here was our motto, Popeye the sailor, who says, "I am what I am." That was good. Okay, here's another one that has been tough. Strabismus. So this is an extremely common ophthalmologic disorder. About 1% of humans has a misalignment of the eyes. This little fellow, you can see the eyes are not correctly aligned. Again, very good evidence for genetic component. Vaishali Parikh, who is a technician in the lab, and I have worked on this for about a decade. Again, only 1 paper to show for it. And even though we collected thousands of samples and had some really amazing families, this was our most amazing. This lady, who I spoke to on several occasions on the phone, had 14 children. 8 of the 14 have strabismus. And they were all seen by the same ophthalmologist in Washington, D.C., Marshall Park, who's really the great father of American strabismus, or really pediatric ophthalmology in general, but strabismus treatment in particular. And we still don't have the gene. So this is one for the next generation. Now, a little more philosophically. It can be hard getting one's work published. And when I saw the poster for this movie, To Hell and Back, I thought this was just perfect. This is about Audie Murphy. For those of a certain age, will know that Audie Murphy was the most decorated veteran of the Second World War. And then he had a career in Hollywood afterwards, and he made this movie about, or maybe they made a movie about his life. The book, To Hell and Back, was the one he wrote. Anyway, publishing is kind of like this, so with a little substitution. And it's not always fun, but you got to do it. Similarly, getting money can be a pain. And I've often wondered why this is. And then I saw this article, low agreement among reviewers evaluating the same NIH grant applications. That explains a lot. And in fact, in the abstract, it says, results showed no agreement among reviewers regarding the quality of the application in either their qualitative or quantitative evaluations. That explains the data. And I was wondering why that was. And I found this article. So this generalizes to the NIH study sections. This is called Extraneous Factors in Judicial Decisions. I'll just note it was contributed or edited by Daniel Kahneman. You may know that name. He's the psychologist who won the Nobel Prize for decision theory. So the study here was in Israel. It was looking at judicial decisions, and the court system that they picked was the parole courts. So the prisoners arrive in the court and they want a petition for parole, and the question is, do they get the thumbs up, yes, you get to get out, or the thumbs down, no, it's back to the jail for you. And the question is, what influences the decision? So what they did was they plotted the time of day when the decisions were made versus the decision, and on the plot are the food breaks. And here's the data in aggregate. So here's time, the beginning of the day, the end of the day, and the dashed lines are the breaks. This is lunch break, and this is the afternoon coffee break. And here is the probability that you get to go on parole. I think you can see the pattern. Perhaps this explains what's going on with the NIH study sections. So the key is get your grant reviewed right after breakfast, or right after lunch, or right after the coffee break. Now, the big question. Why do this stuff? Why, oops, maybe the lights. Oh, someone probably hit the light. That's all right, doesn't matter. It's the last slide. Why do biomedical research? Well, the first answer, I think we all agree, to improve human health. And the second answer, I think, is a huge motivator, to discover something new and beautiful. But I would argue that the third motivation is at least as important, and that's to deepen our societal commitment to rational inquiry. And if anyone needs to see the effect of that in action, there's no better point of evidence than today's public arena and the political world. And we desperately need this. And I'll just give you one example of an experiment that truly embodies the second and third points. This is one of the most beautiful experiments of all time. It was conducted by an undergraduate at Cambridge University. His name was Isaac Newton. And Newton drilled a hole in the wooden shutter of his dormitory room to admit a beam of light, and he passed it through a prism, and he saw that it split up into the component colors, each of which could be purified by allowing it to pass through a little hole in a screen that would otherwise block off the light. And based on that purified lights, he could mix them in various combinations. He could look at their properties. I'll just tell you one simple experiment he did. Now, remember, he's 20 years old. He's in his dormitory room. He has about $2 worth of equipment. But he put a piece of black paper over here, down below the region where there was any light. So nothing to be seen. There's no light there. Put a piece of black paper and then felt it after a few minutes. Anyone guess what it felt like? It was warm. Yes, it was a discovery of infrared radiation. That is, there's a kind of radiation that we can't see. And this, of course, was the discovery that the electromagnetic spectrum is huge. We're just seeing this tiny little sliver in the middle, but now we know, of course, it goes from gamma rays to radio waves. And again, it's just, it was a few minutes in a dormitory room. So this, I think, certainly satisfies the aesthetic beauty part, and I think it also satisfies the rational part. It just took thinking rationally about the universe and discovering something beautiful. So with that, I'll end, and thank you very much.